Good morning, students. We are going to discuss in today's class about the electroanalytical techniques. Electroanalytical techniques basically deal with finding out the analyte concentration by different methods. Now, first of all, let's discuss what is electroanalytical techniques and how does it, really matter, and what is its significance actually. So, electroanalytical techniques basically use the measurement of potential, charge or current, any one of them, to determine the analyte concentration. See, we have already, you are well acquainted with how you have determined the analyte concentration using our basic techniques like titration, correct? Which is where you use a visual indicator. We can determine an analyte concentration using a, a titration where we are using a visual indicator. But what problem comes is sometimes we don't have for every chemical the visual indicator available. Then what to do at that time? That time we can use the potential or charge to characterize their concentration, their reactivity in different ways. Okay. Now it can be a qualitative as well as quantitative method. As you can see on your screen, the slide is showing you. So depending on the analysis, you divide them into various techniques, and the 5 major techniques which are used are potentiometry, voltammetry, coulometry, conductometry and dielectrometry. Out of these, we are going to discuss the 3 major techniques: potentiometry, voltammetry and conductometry as per your syllabus. So now, first of all, before discussing and going in great detail of it, let's try to understand what does this application basically means. What are their applications? For obtaining the thermodynamic data, as I can show you this for the reaction, to generate an unstable intermediate such as a radical ion, okay, and to analyze a solution for trace amount of metal ions or organic species. Now, to generate an unstable intermediate and a radical, we can study the kinetics also during these reactions. Let's understand how. First of all, let's go for the bulk techniques which are available. The bulk techniques basically use conductivity of the ions which are available in the solution. So an example is conductometry, where you do the property of the solution by conductivity of the ions, dissolved ions. Then the next is interfacial techniques, in which the potential, charge or current depends on the species present at the interface between an electrode and the solution in which it sits. It basically deals with pH metry. You might have heard about it, how the pH or H plus ion concentration changes, and we try to analyze the concentration of an analyte accordingly. Now, despite all difference in instrumentation, all electrochemical techniques share several common features. Let's try to understand what common features they share. The electrochemical techniques determines the analyte's form at the electrode surface. We are taking an electrode here, obviously one is positive and one is negative. So the electrode potential determines the analyte form only at the surface of the electrode. So the electrode surface area really matters in this case, how it is reactive. If it is a layer of oxidation, if it has undergone oxidation, we need to check whether the electrode is properly working or giving us the proper reading. The concentration of the analyte at the electrode surface may not be the same as its concentration in bulk solution. So basically we try to take most of the time dilute solutions. That's why, because if you take a concentrated solution, this error will be too much, which is at the surface and the bulk. Third point is correct. Current is a measure of the rate of the analyte's oxidation or reduction. And we cannot simultaneously control current and potential. What does this mean? The last point means is either in all the techniques or most of the techniques, what you try to do is either you keep current constant or you try to keep potential constant and vary the current. Then only we try to estimate the concentration. Now these interfacial electrochemical techniques are divided into 2 types: static and dynamic. What all we do in static, we are trying to understand. Static is that the current is not passed through the analyte solution. Example is potentiometry, in which we measure the potential of an electrochemical cell under the static condition. It is one of the most important quantitative electrochemical methods. Dynamic techniques, in which we allow current to flow through the analyte solution. We are putting the current here. So we are keeping the potential constant, obviously. In the first case, we are not allowing the current to pass. Current is constant. So potential is varying. So it comprises of largest group of interfacial electrochemical techniques. For example, coulometry, in which we measure the current as a function of time. Amperometry and voltammetry, in which we measure current as a function of fixed or variable potential. So in one case, we are measuring current as a function of time. In another case, we are measuring current as a function of potential. This is the list or a flow chart for various electroanalytical techniques which are available. You need not to, go in detail of every technique, but yes, you should know the basic definition of each and every technique in case it comes for a short question to you in the exam. Okay, so electrochemical methods, basically two: interfacial methods and bulk method. Bulk is conductometry or conductometric titrations. Interfacial is static and dynamic. In case the current is zero, it is potentiometry or potentiometric titrations. Potentiometry means you are just calculating the potential of that particular solution. And if you are titrating it against in between a base and acid and trying to understand the end point of this, then it is potentiometric titration. Under dynamic method, your current is greater than zero and you can work under controlled potential or constant current. In case you are working under constant current, then it is coulometric titrations and electrogravimetry. In case you are working on controlled potential, then it is constant electrode potential, voltammetry, amperometry and electrogravimetric techniques. Okay. Now, this again tells you the same along with how they are plotted actually, what graphs you get. So this is just for your information. So we can go with the next slide. Then the second electrode, which we call the counter electrode, completes the electrical circuit and provides a reference potential against which we measure the working electrode potential. So the first electrode is your main electrode, working electrode, and the second is your counter electrode against which completes the electrical circuit and provides a reference potential. Ideally, the counter electrode potential remains constant. So basically, this electrode is a standard electrode, which you sometimes say. And the examples of standard electrode is standard hydrogen electrode, platinum electrode. So all these things you can say. Then electrochemical measurements, which are made in an electrochemical cell consisting of two or more electrodes, and the electronic circuitry for controlling and measuring the current and the potential. So the simple is consisting of two electrodes. One is sensitive to analyte concentration and is called the working or the indicator electrode. If the counter electrode potential is not constant, we replace it with two electrodes, a reference electrode whose potential remains constant and an auxiliary electrode that completes the electrical circuit. Because we cannot simultaneously control the current and the potential, there are only basic experimental designs. Now how we go for it? Measure the potential when the current is zero. Measure the potential while controlling the current. Measure the current while controlling the potential. These are the three ways we can go for this experiment. Each of these experiments designed relies on Ohm's law. Ohm's law you might have studied in your 12th, V equals to IR. Here V is shown with E, so E equals to IR. Now each of these experimental designs use a different type of instrument. Let's try to understand what it is. Now types, potentiometric methods. First of all, let's discuss this instrument and its method. It measures the potential of a solution between two electrodes. The potential is then related to the concentration of one of the analytes. The cell structure used is often referred to as electrode, even though it actually contains two electrodes, an indicator electrode and a reference electrode. So the complete setup is called as a cell structure. Potentiometry usually uses electrodes made selectively sensitive to one ion of the interest. Okay, so that means that particular ion of interest is there, such as fluoride selective electrode. The most common potentiometric electrode used is basically used in pH-metry, where we do the H plus ion concentration, and they are basically the glass membrane electrode. Voltammetry method, it is based on the constant or varying potential at an electrode surface and measures the resulting current with a 3-electrode system. Voltammetry, with its variety of methods, constitutes the largest group of electrochemical methods of analysis and commonly used for the determination of compounds in solution. Now, this voltammetry we can do in various ways, where we can consider amperometry, polarography, etc. Nowadays, polarography is a little outdated because it uses mercury as a electrode, one of the electrode, which is poisonous. We can stick to amperometry majorly. Now, let's first try to understand the basic definition of the next method, which is called coulometry method, which is based on the measurement of the amount of material deposited on the electrode in course of an electrochemical reaction in accordance with Faraday's law. First of all, let me make a very clear distinction that there is one more method which is called colorimetry, which is a spectrophotometric method. It says it identifies the color of the compound and accordingly gives you the absorbance. This is different. This uses Coulomb's law. Faraday's law, Faraday gave to Coulomb's law, where the amount of electricity passed decides the amount of substance deposited on the electrode. The same thing we do here and try to understand the concept. Coulometry used applied current potential to completely convert an analyte from one oxidation state to another. Now, in these experiments, the total current passed is measured directly or indirectly to determine the number of electrons passed. Knowing the number of electrons passed can indicate the concentration of analyte, or when the concentration is known, the number of electrons transferred in the redox reaction. So we are going to wind up here. Then in the next class we are going to start with conductometric methods, where we are going to discuss their titrations and further in detail the whatever definitions we have discussed till now about those we are going to discuss in the next video lecture. As of now, thank you students. Whatever are your doubts, please put it in the comment section.